The modern cell is a very complex system consisting of many parts which perform very specific functions. We've learned a lot about how the cell works in the last 20 or 30 years, but there's still more questions to be answered. One of these questions is how exactly the initial cell emerged and evolved in the first place. And one of the hypotheses is that a primitive compartment, or otherwise known as a protocell, was the first form that eventually evolved into modern cells. This leads us to origins of life research, where we focus on how the chemistries available on early Earth, also known as prebiotic chemistry, assembled into the first compartments on Earth, and how these compartments evolved into modern cells. Compartments in particular are very crucial to the origins of life, as they perform a number of very essential functions to a primitive system, such as concentrating and segregating reactants or protecting molecules from degradation. One process that allows primitive compartmentalization is liquid-liquid phase separation. This process results in the formation of membrane-free droplets similar to colloids or other soft matter, and these droplets can allow compartmentalization and exchange of molecules between phases. In fact, phase separation is present in biology, where a number of membraneless organelles exist in our modern cells, such as stress granules or chromatin. One of the systems that we like to study as model systems that are liquid-liquid phase separated are called complex coacervates. These structures are formed from the binding of oppositely charged biopolymers, such as a positively charged peptide and a negatively charged nucleic acid. And the result is a membraneless droplet concentrated in both of these phases. Coacervates actually perform a number of very important primitive functions, such as the ability to concentrate biomolecules, such as nucleotides or nucleic acids. Coacervates have also been shown to grow and divide through external forces, such as physical agitation or electric fields. Coacervates can have their structural complexity increased through the assembly of multi-phase compartments, each with very specific chemical affinities. And also coacervates can scaffold membrane assembly, suggesting perhaps one transition state between a membraneless and a membrane-bound primitive compartment. In our studies, we focused on another way to potentially increase the structural complexity of coacervates, and this is through the incorporation of DNA liquid crystals. DNA liquid crystals are formed when short duplex oligomers bind together and aggregate end-to-end, forming these long stiff rods. These stiff rods actually will bundle together, forming these nice patterns that can be observed in polarization microscopy. And we sought to combine this liquid crystal structure with a coacervate structure through the addition of a cationic peptide, polylysine. Indeed, we noticed that through addition of polylysine to these DNA structures, we were able to find the co-localization of liquid crystal structures seen below and the membraneless droplet structure seen above through the different types of microscopy. This structure is likely caused by the end-to-end stacking interaction of these DNA duplexes interacting with the polylysine peptides. So we actually were able to find these structures, but they actually only exist under very specific salt conditions. Here, we show that the structure of these liquid crystal coacervate structures, as we call them, is modulated through the modulation of sodium chloride. So for example, at lower concentrations of sodium chloride, what we see are precipitate-like structures. At 800 millimolars of sodium chloride, we see co-localization of the liquid crystal coacervate structure, and through increasing sodium chloride, we see first the disassembly of the liquid crystal structure, followed by the disassembly of the droplets themselves. We believe that this property is due to the fact that increasing salt concentration will help to shield the interactions between the cationic peptide and the anionic DNA backbone, suggesting that higher salt concentrations result in less interaction between those polymers, and lower salt concentration results in stronger interaction between those polymers. We also did an experiment where we engineered a DNA duplex, but with 2 base overhangs. And so this structure actually resulted in the formation of coacervates, but no liquid crystal structures. And we believe that this is because the 2T base overhang actually inhibits the stacking of the DNA so that the liquid crystal structures cannot form. We next wondered whether physical processes on early Earth could have also modulated the structure of these liquid crystal coacervates. And here we subjected the liquid crystal coacervate structures to dehydration, a process that could have been affected on early Earth through, for example, evaporation caused by seasonal or day-night changes. So here we see from A to E, first the existence of a precipitate-like structure. This precipitate-like structure transitions into a liquid crystal coacervate structure shown in B and C, and slowly, through further dehydration, the liquid crystal coacervates, the liquid crystals start to disassemble, resulting in only coacervate structures in D. And finally, in E, we see that the entire coacervate structure has been disassembled. Here is a video showing the similar process. And we believe that this process is due to the fact that the dehydration actually results in up concentration of the sodium chloride in solution, such that by increasing the sodium chloride in the solution, again, similar to what we saw in the salt titration experiments, we see that the system transitions from precipitate to liquid crystal coacervate to non-liquid crystal coacervate to a uniform phase. Next, we wondered whether the system again could be modulated through various other primitive processes that would have been plausible. And so here we study how heat can affect the structure of the liquid crystal coacervates. So we first started at 20 degrees. Upon bringing the system to 50 degrees, we see very little structural change. But upon bringing the system up to 60 degrees, we see a transition from the precipitate-like structures to a co-localized liquid crystal coacervate-like structure, seen here. And upon further increasing the temperature to 70 degrees, we see the liquid crystal structures disassembling, resulting in only the non-liquid crystal coacervate droplets. We next decreased the temperature, and so here we decreased the temperature to 50 degrees, and no significant changes were seen. Upon decrease to 40 degrees, we noticed that the liquid crystal coacervates started to reappear, and they were coexisting with the non-liquid crystal coacervates seen here. And so here in blue, it's one type of liquid crystal, the nematic phase, and in red, it's another type of liquid crystal, the columnar phase. Upon further decreases in temperature, we see more and more appearance of these liquid crystal coacervate structures. And finally, decreasing the structure down to 10 degrees, we actually see a majority of these structures being liquid crystal coacervate structures. What's most interesting about this system is in fact that at very low temperatures, before and after a heating and cooling cycle, the structure is very different. For example, at 20 degrees before heating and cooling, we see predominantly the precipitate form, whereas at 10 degrees after heating and cooling, we see predominantly a liquid crystal coacervate form. And we believe that this is due to the fact that it's very likely that the liquid crystal coacervate form is the most stable form, and so the system requires a heating and cooling cycle, or some other people also refer to this as an annealing cycle, to find this most stable form. So here's a summary figure showing what we believe is happening molecularly within these liquid crystal coacervates. Upon increasing heat and salinity, we see a transition in the phases. First, in red, the existence of the columnar liquid crystal phase with the coacervate phase, where perhaps the DNA rods in blue are interacting with the peptide strands in red, and the rods have bundled together into hexagonal-like forms. Increasing the heat and salinity results in the slight disassembly of these hexagonal bundles, but still the existence of the rods and the interaction with the peptide, suggesting the co-localization between here the cholesteric phase and the liquid crystal phase. Next, upon further increases in heat and salinity, we see the disassembly of these long rods, resulting in shorter DNA duplexes interacting with the peptides, which results in formation of non-liquid crystal coacervate droplets. And finally, upon high heats or high salinities, the entire assembly disassembles. So the DNA no longer interacts with the peptides, resulting in disassembly of the entire droplet phase. So for those who are interested in learning more details about these studies, here's information about 2 papers that were published last year. They have a lot more details of the experiments that we performed and some of the theory that goes into these studies. So I encourage you to look up these papers if you have more interest in learning some of the details. So finally, I'd like to thank all of the funding sources that helped to make this research possible, including those from Tokyo Institute of Technology, JSPS, Kakenhi, Japan Astrobiology Center, and in particular, this project was a collaboration through the French Embassy in Japan, which was a part of a grant program called Programme Exploration France, which allowed a research travel grant to visit a collaborator's research lab in Paris. So thank you very much for your time and attention. I'm very happy to take any questions anytime, and please feel free to contact me by email if you have any further questions. Thank you very much. Have a good day.